Okay, so we're going to look at protein synthesis. So to understand how proteins are made, we understand that DNA is the master blueprint that holds the code for protein synthesis. And DNA directs the order of the amino acids in the polypeptides. So the sequence of code in DNA provides information for producing the sequence of amino acids in the polypeptide. So a segment of DNA that holds the code for one or more polypeptides is referred to as a gene. The code is determined by specific order of the nitrogen bases in the gene. Recall the nitrogen bases are adenine, guanine, and thymine, and cytosine. The code consists of 3 sequential bases called a triplet. Each triplet specifies a code for a particular amino acid. For example, GGC codes for amino acid proline, whereas GCC codes for the amino acid arginine. Genes are composed of exons and introns. Exons are the part of the gene that actually codes for amino acid. They're the ones that are basically actually expressed when the gene is expressed, so that's why they're called exons. And then the introns are non-coding segments interspersed amongst the exons. So what is the role of RNA? RNA is the go-between molecule that links DNA to proteins. RNA copies a DNA code in the nucleus and then carries it into the cytoplasm to the ribosome. So the RNA carries the code from DNA to the ribosome. All RNA is formed in the nucleus. RNA does differ from DNA. It's single-stranded, for one. It also does not have thymine, but instead uracil in its molecule. And RNA has ribose instead of deoxyribose as part of the nucleotide. There are 3 types of RNA. There is messenger RNA or mRNA. There is ribosomal RNA or rRNA, and transfer RNA, also abbreviated tRNA. So messenger RNAs, the first illustration on the bottom left, it is single-stranded, and it's coded from a DNA template strand. So code from the DNA template strand is copied with complementary base pair, resulting in the strand of mRNA. This process is referred to as transcription, which we'll look at in a bit of detail. And mRNA does maintain the triplet code, but every 3 bases are going to be called a codon on the mRNA instead of a triplet as it's called on the DNA. Ribosomal RNA or rRNA, this is the structural component of ribosomes, which is the organelle where proteins are made. So we call the ribosomes protein factories. Along with tRNA, the ribosomal RNA helps translate the message from mRNA into a polypeptide. And the transfer RNA basically is going to carry amino acids into the complex with the ribosome. There are special areas that contain a specific triplet code, and in this case, this triplet code is called the anticodon. And this allows each tRNA to carry only a specific amino acid. So a specific triplet, if you look at the picture down here, the triplet would be right here, the triplet of 3 bases. And whatever that triplet is specific for the amino acids being carried in on the other end of the tRNA. The anticodon on the tRNA is complementary base paired with the codon on the mRNA at the ribosome, adding its specific amino acid to the growing polypeptide chain. And when it does this process is called translation because we're translating from nucleic acids to amino acids in the polypeptide. So protein synthesis occurs in 2 steps, which we mentioned already. Transcription, where we take DNA information and code it into mRNA. And translation, where mRNA is decoded to assemble polypeptides. The overall process is summarized here. Starting within the nucleus, we have transcription, where we go from DNA and make a mRNA transcript. At this point, we have pre-mRNA, and this is where we have to trim out the introns and splice together the exons to form our mature mRNA. The mature mRNA will then leave the nucleus and attach with the ribosomes and begin making the polypeptide in the process called translation. So let's look at transcription now. This is a process of transferring the code from a gene in the DNA to a complementary base sequence in mRNA. So a protein complex of transcription factors activates transcription by loosening histones from the DNA in an area to be transcribed, thus exposing a segment of DNA. Recall that DNA is normally associated with histone proteins. The histone proteins help to organize the DNA into chromosomes later. So if the DNA is wrapped around these histones, you can't access the code within the DNA. So the transcription factors are going to help promote transcription by, first of all, loosening the histones from the DNA. The other thing that it does is that the transcription factors are going to bind to a special sequence at the start of the gene, and this special sequence is called a promoter. And this promoter is only found on the template strand. Remember, there's two strands of DNA, so one is, the one that's going to be read for transcription is called the template strand. And so this promotes the promoter or the transcription factors promotes the binding of an enzyme called RNA polymerase, which is responsible for synthesizing the mRNA. So it's going to promote the binding of this enzyme at the location called the promoter region. Transcription is going to be broken down in 3 phases. The first one is initiation, where the RNA polymerase binds to and separates the DNA strands. And then we have elongation, where the RNA polymerase begins adding complementary nucleotides to the growing mRNA according to the base pairing rules between DNA and RNA. For example, if the DNA template has an A, so here, what this means right here in the notes is that we're going from the DNA base to the mRNA base as we read it. If the DNA has an A, then the complement would be a U on the mRNA. If the DNA has a T, then we're going to add an A to the growing mRNA because that's the complement. And remember, we don't have T on RNA, so we can't have an A. The DNA can't have a T, so the complement would be an A. And if we have a G in the DNA, then the complement would be a C. And if we have a C on the DNA, then complement on the growing mRNA would be a G. Now, where the DNA and the mRNA are temporarily bonded, this location is referred to as a DNA-RNA hybrid, and it's usually about 12 base pairs in length, the hybrid area. And then 3, the termination. This is where transcription stops when RNA polymerase reaches a special termination signal code. So let's take a look at it here in the diagram. At the very top, we have the initiation here. And here's your RNA polymerase. Here is the promoter region. And so with the help of the transcription factors, the RNA polymerase binds to the promoter and pries apart the 2 DNA strands and initiates mRNA synthesis at the start point on the template strand. And so then we go to the second phase here called elongation. So as the RNA polymerase moves along the template strand, elongating the mRNA transcript 1 base at a time. It's going to unwind the DNA double helix in front of it and then rewinds the DNA double helix behind it. So as it moves through there, it's unwinding it as the mRNA is elongating. And so if we come over here to where we blow up this area to look at what exactly is going on, here you have the RNA polymerase. It's unwinding in front, the DNA, and as it's reading it, here you have the hybrid area right here. So as the RNA polymerase is reading the template strand, it's putting in the new nucleotides, the RNA nucleotides, one at a time, and it's doing it according to those base pairing rules. So on the DNA, if you have an A, then you need to be putting a U there because we can't have a T. If you have a C, then this would be a G coming in, and so on according to the pairing rules. So now we're going to come to the termination portion here, where the mRNA synthesis is going to end when the termination signal is reached. So at this point, the RNA polymerase and the complement mRNA transcript are released. So once we get to the termination sequence, then the polymerase and the mRNA are released. So continuing with this process, we've made the mRNA. At this point, it's the pre-mRNA. So this newly formed RNA, it needs to be edited. So before we edit it, or before we process it, it is called the pre-mRNA. And here's where we have to remove the introns, and special proteins called spliceosomes are going to do that. And what they're going to do is only leave the exons, which are the coding regions, and basically literally splices those exons together after removing the sections of the introns. Now we're going to go to translation, where we're going to go from mRNA to our polypeptide. This is going to be a step of protein synthesis where the language of nucleic acids is going to be translated into the language of proteins, which are amino acids. The process involves these items: mRNA, the genetic code. There's a code followed by these molecules. Then there's the tRNA and the ribosomes, the events of translation that will be discussed, and sometimes this will involve the rough ER, and we'll go over what happens there. So let's start with the genetic code. Each of the 3-base sequence on the DNA, the triplet, is represented by a complementary 3-base sequence on the mRNA. We're going to call that the codon. So really, the genetic code that we have printed right here that you'll see in many biology textbooks is based on the triplet code on the mRNA, which is called the codon. So this chart here shows that there are 64 different possibilities. And so this chart shows codon that has UUU in it, or 3 uracils in a row. Then that would code for the amino acid PHE, which stands for phenylalanine over here. So the definitions for the abbreviations are there on the right. As I mentioned, there are 64 possible codons because we have 4 bases in 3 places. So for each position, there is 4 possibles, so that the calculation comes out to 64 possibilities, which are all represented up here in the chart. There are 3 stop codons, and the rest of the codons do code for amino acids. So you can see the stop codons up there in the genetic code. They are UAA, UAG, and UGA. So when if you get a codon that has those triplets there, or if you get those 3 codons, that's where the termination stops. That's where you will stop and terminate the translation process. So there are only 20 possible amino acids, so this means that some of the amino acids are going to be represented by more than one codon, and that's called redundancy, or the genetic code is said to be redundant. So you can see here we have 3 codons for isoleucine, and we have 4 codons for 6 codons for leucine. So it's possible to get the same amino acid with these different codon. So looking at the role of tRNA. So on the right we have a picture of a DNA of a model of our tRNA molecule, and you can see on one end, on the stem end, we're going to have isoleucine here, or basically an amino acid bonded there on the stem end, and then on the other end we're going to have our anticodon there. Okay? Whenever the amino acid is bonded to the tRNA, we're going to call that an amino acyl tRNA, or amino acyl tRNA. The anticodon is going to be, as I pointed out over here, on the head end of the tRNA, and it has a triplet code. So that triplet code is referred to as anticodon, and so it's going to complement the codon. So this particular tRNA will only go to a codon that it complements it. So in this case, the codon on mRNA, let's say we have our mRNA molecule right here. The only place this tRNA can fit is if on the mRNA we have an A, a U, and an A. So those 2 complement, and this would come in, and this right here is the codon for isoleucine. So this tRNA would bring in an isoleucine. So the anticodon of tRNA will bind only to a codon of mRNA that is complementary. I mentioned that a while ago. So if the codon is AUA, only a tRNA with an anticodon of UAU. will be able to bond, as I showed on the previous slide. Now, ribosomes are going to coordinate the coupling of mRNA and tRNA. So the ribosomes are there to help the tRNA and the mRNA coordinate the way they couple together. So the ribosomes contain one binding site for mRNA and 3 binding sites for tRNA. There is an A site, which refers to the amino acyl. So here, this is where the incoming amino acyl tRNA will bring in the new amino acid. And then there's a peptidal site for the tRNA linked to the growing polypeptide chain. So this is where this site is responsible for producing the peptide bond between the growing chain of the amino acid and the new amino acid coming in. And then there's the exit site, where we have tRNA that's already come in and delivered its amino acid at the exit site. That's where that tRNA now, without an amino acid, will be leaving. So here are the sequence of events in translation. Translation occurs in 3 phases that require ATP, protein factors, and enzymes. So all this stuff would require energy and other proteins and enzymes to help. There is initiation, then elongation, and termination. So looking at initiation, we have basically a ribosome is going to be made of a large and a small subunit, and they're sort of illustrated right here. Here is the small subunit, and then over here this is the large subunit. And so the small ribosomal subunit is going to bind to a special initiator tRNA, and this particular one is going to be carrying the amino acid methionine. So all of our polypeptide chains always start with methionine, since it's the initiator. So the initiator tRNA is going to bond with a small ribosomal subunit, and then it'll bind to the mRNA to be decoded. So what's going to happen here is the ribosome is going to scan the mRNA looking for the first methionine codon, which is referred to as a start codon. So the ribosome that's bonded to the initiator tRNA that's carrying the methionine is going to be looking for this codon, AUG, which is our start codon right here. And if we went back and we looked at the genetic code, you're going to see AUG. It says methionine. So it codes for methionine. This is our start codon. Here is our tRNA coming in bringing in that methionine to start the polypeptide chain. So when the anticodon of the initiator tRNA binds to the start codon, the large ribosomal unit can then attach to the small ribosomal unit, forming a functional ribosome. So it kind of looks like a sandwich at this point. But once the start codon and the anticodon find each other, then this large ribosomal subunit comes in and bonds to the complex, forming the larger ribosome. At the end of the initiation, the initiator tRNA is going to be in the P site of the ribosome, and the A and the E sites are empty. So this right here, this location right here, is the P site on the ribosome. And here you have your A site and the E site, which I mentioned earlier. Now we're going to do elongation, and it involves 3 steps. So the first step is codon recognition. And here a tRNA is going to bind to its complementary codon in the A site of the ribosome. So right here we have a tRNA coming in. It's got its anticodon, and it's going to match with the codon there and bind there at the A site, or the amino acyl site. And then the second step is the peptide bond formation. So here, the ribosomal enzymes are going to transfer and attach the growing polypeptide chain from the tRNA and the P site over to the amino acid of the tRNA and the A site. So let's take a look at what that means here. At this point here, if we look at the first picture up above, we have our chain of amino acids, our peptide, and it is currently attached to a tRNA at the P site. What's going to happen here is we're going to transfer that polypeptide to the next amino acid that was brought in, which is right there. And at the same time, there's going to be a bond formed there, so we're going to form a peptide bond. So that's what's meant by peptide bond formation. And this is why that site gets its name of the P site, because this is where the peptide formation is occurring. the peptide bond. And then we have translocation. And translocate means to move here. So here now, the ribosome is going to shift 3 bases on the mRNA, displacing the tRNA by 1 position. When the tRNA in the A site moves into the P site, the tRNA in the P site moves into the E site, and the tRNA in the E site is ejected from the ribosome. So basically, if we move down 3 steps or 3 bases, at this point, the tRNAs that were in the A and the P site are going to all shift 1 position as the mRNA is moving through the ribosome. And so as they shift, the empty tRNA is going to be ejected or exits from the area. The tRNA that was at the A site moves to the P site, and now we have an empty A site, which is ready to accept a new tRNA or amino acyl tRNA. So that brings, continuing with elongation. So once the A site is empty, a new tRNA can come entering, bringing in its amino acid, and the whole process starts over again. So after a portion of the mRNA is read, additional ribosomes may attach to the promoter to start more rounds of translation. So as the ribosome starts moving along, or literally pulling the mRNA through, so it appears as if the ribosome is moving to the right. And as the mRNA is moving through here, that promoter region was over here somewhere. And so the promoter can now attach a new a ribosome to it in the same manner as we mentioned earlier. And when that happens, you may have more than 1 ribosome on the mRNA. And every one of these ribosomes are translating, forming a polypeptide. So how might that look? Let's take a look at this diagram here. So here we have, up here's where we have the promoter region right here. And you have some other ribosomes currently on there translating. This ribosome here has gotten to the stop codon, and so there we have the release of the ribosome and the release of the polypeptide. But over here we have another ribosome right behind it, another ribosome right behind it, another ribosome right behind it, and they're at different stages in producing the polypeptide. And then here comes another ribosome. So we actually see that this is called polyribosome, and this is an actual electron micrograph magnified 400,000 times, showing several ribosomes on an mRNA strand, and each one of them is translating, and that there are different stages of that translation. Then we have termination. So when one of the 3 stop codons is met on the mRNA, so the stop codon is right here. So once we get to a stop codon like UGA on the mRNA, so when we hit one of the 3 stop codons in the mRNA, enters the A site, translation will end, and part of this termination is going to include a protein release factor is going to enter that site, and that's illustrated here. There's the release factor; it enters right in there, and this release factor is going to cause water to be added to the chain instead of a tRNA, and this causes the release of a polypeptide chain, as well as the separation of the ribosomal subunits and the degradation of the mRNA. So if the mRNA molecule is not useful anymore, it'll be broken down. The final polypeptide product will be further processed by other cell structures into a functional 3D protein. So what is the role of rough ER in protein synthesis? Sometimes proteins are going to be produced in the ER instead of freely in the cytoplasm. So let's take a look at that real quick. A short amino acid segment called the ER signal sequence is going to be present on the growing polypeptide chain, and is going to signals associated ribosome to dock on the rough ER. And there is a signal recognition particle on the ER will direct the mRNA ribosome complex where to dock or connect to. Once docked, the forming polypeptide will enter the ER. So as the polypeptide is formed, it will, instead of the chain being free in the cytoplasm, it's going to enter the ER. And so sugar groups may be added to the protein and its shape be altered while it's in there in the endoplasmic reticulum. The protein is then going to be enclosed in a vesicle for transport to the Golgi apparatus for further modifications and packaging. This is it's probably easier described on this diagram here. So here you have your mRNA, and here's your ribosome. And looking at step 1, the signal recognition particle is going to direct the mRNA ribosome complex to the rough ER. There, the signal recognition particle binds to the receptor site. Then we're going to start growing our polypeptide chain. Okay, so we grow our polypeptide, and polypeptide is going to snake through the ER membrane pore and into the cistern. So it's going to go into the cavity or the space, the fluid-filled space within the ER here. And as we continue to read the mRNA, the polypeptide chain is going to grow. So all this is just showing a sequence of steps as this process progresses. In reality, the ribosome is probably stuck in place somewhere on the endoplasmic reticulum. So looking at step 3 here, an enzyme is going to clip off the signal recognition sequence as the protein synthesis continues. Sugar groups may be added to the peptide chain. So here we have a sugar group being added there. And then in this example, the completed protein is released from the ribosome and folds into a 3D conformation, a process that will be aided by molecular chaperones. So there'll be proteins or molecules within here that will help aid in the folding of this. And then in step 5 here, the protein is going to be enclosed within a protein-coated transport vesicle. And the transport vesicle will make their way to the Golgi apparatus where further processing of the protein occurs. So, in summary, from DNA to protein, we have complementary base pairing, directs transfer of genetic information in DNA into amino acid sequence in the protein. The DNA triplets are coded to mRNA codons, so from DNA triplet to mRNA codon. The mRNA codons are base paired with tRNA anticodons to ensure correct amino acid sequence. So the codon is directing what amino acids will come in, because the codon matches the anticodon with on a tRNA carrying a specific amino acid. So the anticodon sequence of the tRNA is going to be identical to the DNA sequence, except that we have a uracil is going to be substituted for thymine. And while this is mentioned here, may not make much sense, but let's take a look at what that means when we see it in a diagram that simplifies the entire process for us. If we start with a gene or a segment of DNA right here, which is going to be the template strand, the other strand is the one that's not going to be transcribed here. So this is the base sequence all along that strand there. And so we have thymine because this is DNA. And so what we're going to do is we're going to transcribe that. And when we transcribe that, we're going to make the mRNA. So this represents the mRNA sequence that came from the DNA sequence. So with T, the mRNA would have a U. With A, the mRNA would have a U. With C, the mRNA would have a G, and so on. So this sequence here was determined by this sequence here in the DNA. Now these are the codons. Each triplet here is a codon now. Okay? And those codons are going to match with the anticodons for the tRNA. This tRNA here is carrying a methionine. This is the start codon AUG. Remember, a ribosome helps assist with the tRNAs coming in, assist with its coupling, and then we have the next tRNA coming in for the next codon, complementing that codon, bringing in another amino acid, proline, and so on. Remember, the ribosome is connecting these amino acids, and they're coming in a sequence that is based on the code, or the codons, the sequence of these codons as they go in. So every one of these amino acids was purposely put there based on the code. And then you get to your stop codon and it's released, and then here is your polypeptide being produced. So if we go from the top DNA to here, this is a process called transcription. And then this part down here is describing the translation. Looking at the last section of the chapter, covers topics called apoptosis, autophagy, and proteasomes. So here, cells that have become obsolete or damaged, they're old, not needed, or damaged, are going to be needed. We're gonna need to take them out, and so there's a system to remove them. One is called autophagy, and this means self-eating, and is a process of disposing of non-functional organelles and cytoplasmic bits by forming autophagosomes, which can then be degraded by lysosomes. Unneeded proteins can be marked for destruction with ubiquitins. These are proteasomes that disassemble ubiquitin-tagged proteins, recycling the amino acids and the ubiquitin. Apoptosis is also known as programmed cell death, which causes certain cells, example cancer cells, infected cells, and old cells, to neatly self-destruct. This also is actually a part of normal development as well. When we are developing early on, we have webbing between our fingers, and then the cells that make the webbing are programmed to die at some point during our development, which is why we're not born with webs between our fingers. The process, though, begins with mitochondrial membranes leaking chemicals that activate enzymes called caspases. The caspases cause degradation of DNA and cytoskeleton, which leads to cell death. The dead cell does shrink and then is phagocytized by macrophages, which are related to white cells. So all cells of the body contain the same DNA, but not all cells are identical or carry out the same function. So chemical signals in the embryo channel cells into specific developmental pathways by turning some genes on and others off. This process of development of specific distinctive features in cells is called cell differentiation. So this explains how you can start with one cell, and then as the cells divide and many cells are produced, these cells can go down different paths and turn into different cell types, like muscle cells or skin cells or nerve cells. They all have the same DNA. It's just a matter of turning on the right genes at the right time and turning off others at the right time to get these different types of cells that have different functions. So the subsection of cell destruction and modified rates of cell division. Organs are well formed and functional before birth, but we need cell division for growth. So cell division in adults is needed to support or to replace short-lived cells and to repair wounds. So short-lived cells might be our skin cells. So we constantly need to have cell division to replace those skin cells. And when we have damage to tissues, we need to repair them, and that's done by cell division. Hyperplasia then is the accelerated growth that increases cell numbers when needed. And then atrophy is kind of related here, but if cells are not being used or not needed, they're going to decrease in size, which results from loss of stimulation or use. And then what about aging? So why do we get old? What happens when you get old? What happens when you age? So some mechanisms have been proposed for how aging occurs. The first is wear and tear theory. This is a theory that says that a lifetime of chemical insults and free radicals have cumulative effects on the cell or on cells. And then there's mitochondrial theory of aging, and here free radicals within the mitochondria diminish energy production. We recall these radicals that have been mentioned. These are molecules that are highly reactive and toxic to cells, so they go in and cause destruction to parts of the cell. And then another theory is immune system disorder. Here you have autoimmune responses as well as progressive weakening of the immune system. And autoimmune responses are basically when your immune system starts attacking itself, or starts attacking some of the parts or cells of your own body. So that's why it's called autoimmune. And then there's genetic theory, and here we have what's a cessation of mitosis and cell aging are actually programmed into your genes. And so here, you think about the DNA is double-stranded, and so capping the ends of the DNA or these chromosomes are these structures are telomeres, which are strings of nucleotides that protect the ends, kind of like caps on the end of shoestrings, keep the shoestring from fraying apart. So every time a cell divides, a little piece of the telomere breaks off, so the telomere shortens. So the analogy here would be as if the telomere was an hourglass. So every time the cell divides, you lose a little piece. So ultimately, there's a point where the cell can no longer divide because you have to make the DNA, basically the chromosome, falls apart as the ends of the telomeres have worn away. There is an enzyme called telomerase, which is an enzyme that lengthens the telomeres or continues to add to these telomeres, but you don't find this in adult cells. And so this is why the theory as to why we age is that we no longer replace or add nucleotides to the telomeres. Instead, it wears off like an hourglass. However, this telomerase is found in germ cells of the embryos, but absent in adult. And we also interestingly find this enzyme in cancer cells. So cancer cells basically are thought to be immortal.